Hi, I'm Wong Xiaoxian. Today, I would like to present my research proposal. The research title is Dysregulations of Citrullinations by Oral Bacterium and its Effect to Neurodegenerative Disorder, a systematic review. I will be presenting the introductions, literature review, research questions, objectives, research hypotheses, material and methodology, the expected outcomes Gantt chart of this research, and the reference list. Neurodegenerative disorders are considered as the progressive neural system damage and cognitive functions decline. In recent years, the scientists are eagerly studying the pathogenesis of these conditions. One of the emerging findings is dysregulation of citrullinations. Citrullinations is also defined as deaminations. Is considered as stress-induced post-translation modifications of proteins by the conversion of the primary ketamine group to ketone group of the arginine residues. This process is mediated by the actions of calcium ion-dependent peptidyl-arginine deaminase, so-called the PAD enzymes, to convert the amino acid arginine into citrulline. And this process highly requires the calcium ion. This conversion involves the shift in charge, where the positively charged arginine residue is modified into a neutral citrulline residue. Interestingly, the scientists has also discovered the association between the infections by oral bacteria and the dysregulations of citrullinations, initiating the pathogenesis of autoimmune and neurodegenerative conditions. Not only that, the mechanisms of pathways of oral bacteria to trigger the impairment of immune or neural system are the hot topics that are continuously being discussed. In the literature review, citrulline has been concluded as a non-essential amino acid and a product of post-translation modifications on proteins, since the tRNA code for citrulline does not exist. As a result of citrullinations, the citrullinated proteins will affect the protein structures and functions, even able to denature the proteins. As well, the citrullinated proteins become the target antigens for anti-citrullinated peptide antibodies (ACPA), and they form the immunocomplex that initiate the complement system. Moreover, to focus on the process of citrullinations, the types, function, and target site of PAD enzymes which initiates this process have also been widely studied. The PAD is claimed to be tissue-specific enzymes. For example, PAD2 can be found in many organs such as the brain, female reproductive systems, skeletal muscles, and hematopoietic cells. PAD is mostly involved in the citrullinations of myelin basic protein, which is responsible in maintaining the stability and structures of myelin sheaths. Therefore, there were studies conclude that PAD2 was found to be an important trigger point of the pathogenesis of neurodegenerative disorder, namely the multiple sclerosis. In the review of associations between the citrullinations and neurodegenerative disorder, frequent protein citrullinations is a hallmark of neurodegenerative disorder. They are caused by myelin protein aggregations and reactive astrogliosis. For example, amyotrophic lateral sclerosis, Alzheimer's disease, Parkinson's disease, and multiple sclerosis. Furthermore, the gram-free acidic protein, GFAP, which is mainly located in astrocytes, is a substrate bound to human PAD2 and initiates the citrullinations of GFAP. Accordingly, it leads to the progression of Alzheimer's disease, and autoimmune disease. Now, let's proceed to the association between oral bacteria and citrullinations. For Pheromonas gingivalis is a non-oral bacteria that produces its own peptidyl-arginine deaminase, which is called as PPAD. PPAD has the ability to catalyze both human and bacterial proteins without the requirement of calcium ions. Consequently, the citrullinated proteins consistent the target antigens leads to the production of ACAP and initiation of autoimmune reactions. The infections by Pheromonas gingivalis triggers the formation of neutrophil extracellular traps, which is able to damage the periodontal tissues and recruit more active proteins and PADs. This process further drives the citrullinations and production of ACAPs. To study the association between oral bacterial and neurodegenerative disorder, for Pheromonas gingivalis produce gingipain R, RGP, which is a cysteine proteinase. It degrades the tau protein structures and leads them into proteolysis, causing microtubule destabilizations, reducing the effectiveness of axonal transport, and eventually the neural system is degenerated. As well, for Pheromonas gingivalis can release outer membrane vesicles, which is enriched with RGP, are able to internalize themselves into the host cells, initiating inflammasome activations in microglia and the formation of apoptosis-associated spike-like protein space that triggers beta-amyloid assembly and deposition, potentially forming either soluble amyloid fibrils and amyloid plaques. The research questions are: what are the molecular pathways and oral bacteria involved in these regulations of citrullinations in the human body? And do bacterial periodontal disease increase the risk of neurodegenerative disorder? The objectives of this research are to determine the molecular pathways and associated oral bacteria disregulating the citrullination process in human body, and to assess the credibility of evidence supporting the associations between oral bacterial disease and neurodegenerative conditions from meta-analysis and observation studies. The first research question is not hypothesis driven, while the non-hypothesis for research questions 2 is bacterial periodontal disease are not statistically significantly associated with the increased risk of neurodegenerative disorder. And the alternative hypothesis is bacterial periodontitis periodontal disease are statistically significantly associated with the increased risk of neurodegenerative disorder. Next, I would like to introduce the materials and methodology in two parts, as the research is planned to be in two stages study. The stage 1 study is to conduct a systematic review about the molecular pathways and oral bacteria contributes to the dysregulated citrullinations process, while the stage 2 is to study the associations between the oral bacterial disease and neurodegenerative disorder and assess the credibility of evidence. For stage 1, the first stage is a comprehensive literature search will be conducted across multiple electronic databases. The systematic reviews will include in vitro studies published in English for exploring the molecular mechanisms and pathways involved in the dysregulation of citrullinations by oral bacteria. The exclusion criteria in study selections are the studies that do not specifically emphasizing the role of oral bacteria in citrullinations and are not related to environmental mechanisms relevant into human health, and non-peer-reviewed articles, editorials, commentaries, and opinions pieces. Next, the data will be extracted from the standard studies using a standardized data extraction form, including the study characteristics, molecular pathway, and molecular pathway to dysregulate citrullinations and identifications of oral bacteria. Then the quality of the included study will be assessed using TOPS tool and will be rated as high, moderate, or low quality based on their methodology and risk of bias. In step 3, the comprehensive overview of the molecular mechanisms and associated oral bacteria dysregulating the citrullinations will be evaluated. For stage 2 study, the electronic sources will be used to identify relevant articles, peer-reviewed systematic reviews, and meta-analysis of observational studies with case control or cohort studies measuring any associations between periodontal disease and neurodegenerative disorder or the related outcomes in any populations of any age will be included. After that, the data extraction will be done independently by 2 reviewers, and in case of discrepancies, a third reviewer will be consulted. To grade the methodological quality of each meta-analysis, AMSTAT version 2 will be used. For each association, meta-analysis will be re-performed again to calculate the adjusted summary estimate and corresponding 95% confidence intervals with p-value using the random effects models by Di-Simonian and Large method. In each meta-analysis, heterogeneity will be evaluated using the I-squared statistic. The evidence of small study effects will be assessed by Eagles test. We will also conduct the meta-analysis again using a series of credibility series from 0 to 30%. The strength of the evidence per association will be graded according to the amount of evidence, statistical significance, heterogeneity, small study effects, assess significance, and credibility settings. The sensitivity analysis will be performed also for association initially graded as having class 1 or class 2 evidence. Also, we will approach for the meta-analysis with fewer than 5 studies. As well, subgroup analysis stratified by study design and the type of bacterial disease will be performed as well. In the end of this research, the specific molecular pathways used by the study oral bacteria to influence the citrullinations process in human body should be identified comprehensively with reliable scientific and statistical analysis and grading. Meanwhile, the characteristic of mediated proteins and enzymes employed during the process of human citrullinations can be well identified at the end of this study. Not only that, in order to answer the proposed research questions, a convincing justifications on the associations between the oral bacterial infections and neurodegenerative disorders should be concluded together with the findings on the credibility of evidence per associations. The combinations of these outcomes are expected to elucidate the link between oral bacterial infections and central nervous system impairment. As well, this study should illustrate the importance of oral hygiene to prevent the onset of neurodegenerative disorder if the association between the oral infections and neurodegenerative disease is confirmed. This is the estimated budget to be used, and the Gantt chart will be applied in this research. And this is the reference list I have used. This is the end of my presentation. Thank you.